Alrighty, hello everybody. We are back. Today we're looking at lecture 5. We're going to look at breaking and repairing the DNA phosphate backbone. In other words, this lecture is on restriction enzymes and ligases, are things that you have probably seen in Bio 200. Well, ligases you've seen in Bio 200, but restriction enzymes not so much. So, to start with, I wanted to go over this. I believe this was a practice question that was given, but this is kind of one of the weird questions that we kind of see in biochem. Is that, like, you'd think that um, double-stranded DNA would have a higher melting temperature than double-stranded RNA, but it's actually not true. Double-stranded RNA actually surprisingly has a higher melting temperature, and what that means is that typically at a neutral pH, double-stranded RNA has like a 20 degrees Celsius higher TM compared to double-stranded DNA. So double-stranded RNA is 20 degrees higher in melting temperature than DNA, double-stranded DNA, assuming identical sequence, which is mentioned here in this point here. But it's interesting because double-stranded RNA is definitely more rigid than double-stranded DNA. Now, I remember I said that single-stranded RNA is a lot more flexible than double-stranded DNA. That's still generally true. I mean, single-stranded RNA is, it's a lot more flexible, right? I mean, it's single-stranded. It's flapping around a bunch. DNA is locked in place in comparison. So that comparison is still there. But between double-stranded DNA and RNA, a double-stranded RNA is for sure more rigid. So in terms of like this expression that we've seen from biochem now at this point, and from gen chem and all these things, being more rigid for double-stranded RNA means it's more entropically unfavorable. But the fact that it has a higher melting point means that it must be very enthalpically favorable. So this delta H term must be so negative that even though the delta S term is negative as well and it's unfavorable, it doesn't matter. The delta G is still very negative. So what makes it so that double-stranded RNA is more stable, even though it's in the A form, is that the base stacking in the A form makes it more enthalpically favorable, and thus more stable than double-stranded DNA. It's kind of interesting. I thought you'd think that with double-stranded DNA, that would have a better base stacking in the B form, but not necessarily, which is even I'm a little bit puzzled by that. So it's a bit of a weird conundrum, to be honest with you. But I do remember this question. It was definitely a little bit weird. So, yeah. Hold on one second. Sorry, I just realized that like midway through that I forgot to add some or oh no, never mind. Okay, I thought I had downloaded the wrong lecture, but no, we're all good. Anyways, yeah. So yeah, you'd think that like that double-stranded RNA would be less stable, but it's actually not the case, which is really really interesting. And it's the reason actually has to do with in double-stranded RNA, the extra 2' hydroxyl group. So that 2' hydroxyl group with single-stranded RNA is pretty problematic because it could self-hydrolyze. But in double-stranded RNA, that 2' hydroxyl group, it's so locked in place by the fact that it's double-stranded that it actually can't really do any hydrolysis reactions, but it actually allows for extra base stacking and extra hydrogen bonding with like the ribose sugars and all that stuff, that it actually leads to higher thermal stability. So TLDR, the 2' OH actually contributes to stability of the double-stranded RNA, but actually also is what results in the lower stability of single-stranded RNA. We already talked about the different base forms, right? We have the interactions in the double-stranded helix. We've got our hydrophobic van der Waals interactions. And then we've got our base pairing as well. And the phosphate backbone obviously has electrostatic repulsion. So I won't go over that again, but if you want to look at that, lecture 3 is available, or lecture 4 is available to look at. And you've got A, B, and you got the A form, the B form, and the Z form of DNA. So lecture 5, we're going to talk about enzymes that break and join the DNA together. And we're going to start by taking a look at protein binding sites in DNA. So there's actually a lot of places that things can bind. They can bind to the positive charges could bind to the phosphates here. Maybe some polar stuff could bind to the phosphates. You could also maybe try and have something interact with the bases. Although interacting with the bases directly is a little bit more difficult. And also, the nice thing about this image is it actually shows you really well the major and minor groove. Look at the difference between this space here and this space. That's like the difference that you're looking at when it comes to the major and minor groove, and it actually makes a huge difference when you're trying to make proteins fit in. So it's much easier to fit a protein into this space than it is into this space. So that's why the major groove is where most of our interactions occur. But yeah, you could have like hydrophobic interactions. Maybe the protein can bind into this area and like interact with the bases. The positive charge obviously on the phosphates and the polar uncharge as well on the phosphates. So there's plenty of amino acids that can interact with DNA when it comes to proteins. So proteins can interact with DNA in a lot of ways. They can hydrogen bond. They could have base stacking, like the tryptophan right here. So there could be a tryptophan right here and a tyrosine here. So there could be some base stacking that goes on. You could have some arginines, right? So plus minus interaction. This is a phosphate right here. You could have some electrostatic interactions, more electrostatic interactions, lysine, histidine, more electrostatic interactions, lysine and phosphate, histidine and phosphate, and then more stacking. So there's a whole bunch of interactions you could have. Right, there's stacking, there's hydrogen bonding, there's more hydrogen bonding, there's electrostatics right here. There's an electrostatic interaction, or actually it's a hydrogen bonding interaction. But I mean, there could be. I guess this would maybe be electrostatic, more base stacking. There's a whole bunch of things that could happen. So all this to say that there's a whole bunch of different possible interactions that DNA could have with proteins, and that just comes as a result of the structure of the protein and what amino acids it has. So some of the protein motifs that can fit really well into grooves, as we learned in 440, are there's alpha helices and there's beta sheets. Alpha helices fit great into DNA. Actually, in 442, you guys talk about zinc fingers, which is like a way of editing DNA. That's actually one of it uses a helix, an alpha helix, to actually edit the DNA, and it fits into the major groove, not the minor groove. Minor groove is too small. A good fit would be a two-stranded beta sheet, so it would just be like I believe it would just be like this, if I'm not mistaken. Because you can have beta sheets like this, right? But those will be too large. So a two-stranded beta sheet fits pretty well as well. And you can see that sometimes you can coordinate waters and stuff. Like you can coordinate waters to do some H bonding and stuff like that. You could also the protein could directly act. So you can see here we have some amino acid that's directly interacting with the bases. Or like I said before, maybe a protein coordinates some water molecules, and those water molecules actually interact with the DNA. So the whole bunch of different possibilities. The other thing that I want to kind of mention is the surface charge distribution. So when you think about proteins and binding proteins, right? DNA is a very negatively charged species, we'll call it, or a negatively charged molecule. And in order for proteins to bind to very negatively charged molecules, what we need to do is we need to have a very positive protein, and at least in the binding site, specifically for the DNA. And that's actually exactly what we see. We see that, especially in the binding pocket right here, very, very positive, extremely positive. And that makes perfect sense. Like all these phosphates, they need to be interacting with the protein, right? And in order to keep it stuck in place so the enzyme can actually do its job. So it makes sense that the protein's active site would be a very positive place. And, abrogates is a very funny word. I had to look this up when I first saw it. I kind of figured I could, reason through it, but, still, it's kind of weird. Abrogates means that it, deletes, or, it, reduces the ability of something to do something. So, a restriction enzyme, which is what RE stands for, restriction enzymes are basically cleaving enzymes. They basically cut DNA. And what this is basically trying to say is that DNA methylation, so adding a methyl group to DNA, makes it harder for restriction enzymes to cut. That's all it's saying. It's also possible that methylation actually increases the ability for restriction enzymes to cut. It just depends on the type of methylation and the type of restriction enzyme that you're looking at. It's important to also note that methylation generally occurs on these CPG islands. That's more important in 442, but it always occurs in these CG pockets. And it does occur elsewhere as well, but that's the main thing that we know so far in terms of where methylation occurs. So all this to say that, DNA methylation, so modifications of bases like methylating cytosine, actually leads to changes in how proteins interact with the DNA. So if you want to stop a DNA molecule from being cleaved by a restriction enzyme, methylate it. And if you want something to actually start cleaving, if you want to actually cleave something that's methylated, you can demethylate it and actually start cleaving it. So, moving on to restriction enzymes, let's just have a bit of a chat on, how these things work. So there's a couple ways you can describe it. So nucleases, right, is just a fancy way of talking about things that cleave, if I'm not mistaken. But they specifically cut DNA and RNA, and they do this by breaking phosphodiester bonds. That's what a nuclease is. An exonuclease starts from outside on the ends of the DNA and works inwards. So, if I have my DNA molecule like this, right, the exonuclease would start on the very ends of the DNA. It would start, on this side, or it would start on this side of the DNA, or RNA. That's different from an endonuclease, which I'll talk about in a second. And, it chews away, the exonucleases, they chew away at the free end, like the 5' end, right? It chews away at the 3' end. It chews away at the ends. It starts from the outside and goes inwards. And we have different ways of classifying these nucleases, and the way that we classify them is whether or not it chews DNA or RNA. If it chews DNA, we call it a DNase. If it chews RNA, we call it an RNase. And the direction of the chewing is actually it could go both ways. You could have a 5' to 3' direction for the chewing, or there are some nucleases that do 3' to 5'. So, different types of nucleases. And they can, it doesn't matter. They're not very specific either. So, unlike restriction enzymes, which we'll talk about, endonucleases, we'll talk about in a second, these are not very specific. So, they just kind of start from the outside, go inward. It does not matter. There's no specificity. They'll just start from the outside. They don't care what's on the outside, and they'll chew until they reach the inside. But endonucleases are a little bit different. Endonucleases cut in the middle of a strand of a DNA, or a duplex, or whatever you want to call it. So they cut in the middle, and similarly they also cut DNA and RNA. So similarly, it's a similar thing. But unlike exonucleases, they can be non-specific or specific. So they can be either one. So there are some specific enzymes that we call restriction enzymes, and these are endonucleases that cleave only at specific DNA sequences. And that's what we call restriction enzymes. So there are different types of endonucleases. There are non-specific endonucleases, and there are specific endonucleases. And the ones that are specific are what we refer to as restriction enzymes. So just a real quick breakdown of how restriction enzymes work is they're endonucleases, so they start from the inside of DNA and they cut out. And the way that they actually decide where to cut is that most restriction enzymes have to identify a palindromic sequence. And a palindromic sequence is when you can read a section of DNA the same way forward on one strand as you can on the other strand. So if you see here, they haven't really 5' 3' here, but if I read this bottom strand from 5' to 3', it's GAATTC. And if I do the same thing for the top strand at that same box, it's also GAATTC. That is a palindromic sequence. The bottom strand forward read is the same as the top strand forward read. So that is what a palindrome is. And what that signifies to the restriction enzyme is that is an area that requires cutting. And generally speaking, when restriction enzymes cut, they cut in this like zigzag pattern. So it leaves this like overhang here, like this 5' overhang. So that's a specific kind of thing that restriction enzymes do. And you don't have to memorize these. By the way, this is just something that is nice to look at, but these are all different restriction enzymes. You can see that they all cut in a very similar manner where they leave these overhangs. So you may be wondering, how does this kind of cleavage work? And the answer to that is, well, they just use water. Basically what happens is you can think of it like we deprotonate this OH, this water that comes in, and then we're just going to use the lone pairs on this water to go like this. We're going to attack that. This is going to break off, or not that, and then this is going to break off, and then you're left with this and this. And restriction enzymes, they tend to have magnesiums in their active sites, and this helps activate this water molecule. Like I said, it helps kind of like deprotonate it, if you will. Not necessarily deprotonation, but it helps with positioning more so than deprotonation. But it helps position that water molecule so it's in a place where it's much easier for it to attack the phosphorus. And it does this by stabilizing the phosphorus negative charge, and by putting the water in the right place to actually attack that phosphate group. This is just an example of a restriction enzyme. You don't have to memorize this, I don't think, but the EcoRV is a restriction enzyme. It interacts with the major groove, important to keep in mind, by hydrogen bonding. And it does so through a couple glycines and an asparagine. And that's only if it's the cytosine and guanine, but then if it's thymine and adenine, it interacts through threonines and asparagines. So yeah, that's just an example, and it kind of just goes to show that, it uses a palindromic sequence to cut, and this is how it kind of binds. This is the mechanism it uses to bind with guanine and cytosine, and this one for thymine and adenine. Methylation of the host DNA prevents restriction enzyme binding. This is just another way of saying, it's kind of like if you're driving on a road and then someone puts a traffic cone in front of you or like a blockade in front of you, right? If you're driving along and someone puts that blockade in front of you, you can't drive forward anymore. That's kind of how methylation works. It's like you're putting a blockade in front of the car. So essentially what's happening is, normally this is how the interaction occurs with the restriction enzyme and the DNA, but when you do some sort of methyltransferase reaction and put a methyl group on there, those interactions are no longer there. Like these amino acids can't interact with the DNA anymore, and so it just doesn't interact at all, and then nothing happens. So actually, a lot of bacteria use this method to actually stop themselves from, getting destroyed, right? Because otherwise, they'll go into like and they'll try and invade a host, and their DNA will just get cut up completely because, these restriction enzymes will just cut them. But if they're methylated, they actually can't get cut. So it's a cool little defense mechanism. Um, Moving on to this, I'm not going to spend a lot of time on this. This is basically just him going over how do restriction enzymes find their target. There's a whole bunch of research on, like, oh, do restriction enzymes hop or do they slide? Like, do they slide along the DNA or do they, like, jump from place to place? Long story short, from this experiment, the answer is they do both. They both hop and jump. It just kind of depends on the situation. So I wouldn't even bother trying to go through all of this. The short answer is they do both. So yeah. Yeah. So for here's just a nice little crystallite. There's a crystallization example of how it's non-specific and specific binding is. Again, I don't think this is that important to like. I don't really think that the images are that important for your guys' content. If you guys want to look at these and you want to learn more about these, feel free. But I don't think this is that important. So, the question of can hopping also happen is what we talked about before. I mean, the previous experiment suggested that sliding was one of the main ways that it does it, but hopping does happen. And what you end up finding is that depending on the situation, sometimes it hops more than it slides. So like, see in this case, because it's so open, like this like one here, this like longer one here, because it's so open, it tends to slide along these sections and then hop between these sections. So depends entirely on the structure of the DNA. The last thing that we're going to look at is DNA ligases. And DNA ligases are fun little tools. This enzyme mechanism is something that I recommend you like drill into your head of how it works. And the important thing to keep in mind also is the cofactor. So generally speaking, in eukaryotes, it's ATP. And essentially what it does is it uses that ATP to kind of catalyze a connective reaction where you're connecting two strands together. Bacteria actually use NAD as their cofactor, but in this example, you should be able to see what's basically happening here is we start here with our lysine, and this lysine actually acts as a nucleophile. And when the ATP kind of binds into this spot here, you can imagine there's probably like a magnesium cofactor in here or something that allows it to kind of stabilize in there. This attacks this phosphate, breaks off this pyrophosphate, and that pyrophosphate can leave, right, and get cleaved in two, and that would release like 30 kilojoules, right? That's a lot of energy, so that makes it a very stable reaction. And you then form this intermediate where you have this lysine covalently bonded to the AMP now as it is. From there, you end up getting this thing where the you end up what you do is you bring your DNA in, and so now this is your broken DNA. This is your broken DNA right here, and so you take the 5' phosphate, and that 5' phosphate attacks the phosphate that's attached to the ligase, and then you end up with this intermediate where now the phosphate, right, this 5' phosphate is now linked to another phosphate. And so the last step that happens is the OH, right, because phosphate is actually a pretty good leaving group. The OH on the 3' end actually comes in and attacks, actually technically the 5' end, if I'm not mistaken. Actually, no. No, sorry, this is 3'. The OH on the 3' end of the broken strand actually comes in, attacks that, and kicks off that O, and then you're left with a functional piece of DNA. I just messed up the freaking thing. Uh-oh. But yeah, then you're left with a functional piece of DNA. So I hope that makes sense. Like, you start with this, you covalently attack, right? This kind of gets split off, right? And then you release this PPI, turn it into 2PI, and then that's pyrophosphate, right? Very energetically favorable. Then you have this enzyme AMP complex, where like this lysine is directly connected to this AMP molecule. The conformational change happens, the DNA binds, and then it does a nucleophilic attack, where it basically grabs this phosphate from the lysine, and then from there the 3' end of the DNA comes in and attacks, and it completely reforms the bond again. This is just a nice little slide that talks about restriction enzymes and ligases. Not very important, I don't think. Essentially what you can do is you can actually use ligases and restriction enzymes to cut out portions of DNA from bacteria and insert your own your own gene of interest. Something you actually do in the Biochem 426 lab. But you can essentially take this take a plasmid, cut this part of the plasmid out, right, like seen here, insert your own gene of interest, transform it into E. coli or bacteria, and then have those E. coli replicate the DNA, and stuff like that. And then like this whole process here of cutting this out and cutting out this section and pasting this yellow section in requires restriction enzymes to break it, first of all, and then ligases to actually put the yellow part in there. So this would require an RE, and this would require right here, this would require a ligase. So, yeah. A shorter video today, I think. But I hope you guys enjoyed this video. I hope it was helpful. On the shorter side, but I don't think I need to go too deep into the weeds with these lectures. Just trying to make sure that I reiterate the important points of the lectures. So, if I missed anything, please let me know. But other than that, thanks for watching, and I hope you have a good day. See you later.